All righty, it looks like we're ready to roll. Well, hello and good morning, everyone. Thank you so much for hopping on to today's webinar. My name is Kendra Wagner. I'm going to be co-host today. I'm a research scientist with Rainbow Ecoscience. And today we're going to be talking about beech leaf disease and getting an update on latest management protocols. Our main guest today is Mr. Mark Ware. He's our arborologist with Rainbow Ecoscience, and he's going to be filling us in. Before we hop off, or sorry, before we really get started, the chat is disconnected. If you have any questions, please put them in the Q&A. Any questions or comments or concerns, drop them in the Q&A and we'll be able to see them and answer them afterwards. So again, the chat is offline, it's disconnected. Please put anything in the Q&A box. And with that, we'll get started, Mark.
All right. Well, thanks, Kendra. Let me share my screen here. And all right. Can you see that all right?
I can.
Excellent. Well, good afternoon or morning, depending where you are. Exciting topic today. Some of you may be very familiar. Some of you this may be new to, but we are going to be talking about beech leaf disease today. We're going to go through a bit of the biology, take a look, a very high-level look at some of the most recent research that's come out around this, as well as get into a couple management options that we have for it. So exciting stuff that we're going to get into today. As Kendra mentioned, my name is Mark Ware. I am an arborologist, which is a fancy, slightly made-up term for, I do a lot of technical support. I get to assist the research and development team on occasion, which is always fun. Get to go out with clients and get some hands-on training with our protocols and products and equipment. And so it's a pretty unique role and one that I enjoy doing. As far as Rainbow Ecoscience, most of you are probably at least somewhat familiar with us, but these are some of the products and pieces of equipment we offer. We'll be talking about a couple of these specifically later on. And then last but not least, why are we talking about beech leaf disease? Well, it's obviously a big deal in our industry right now. And as part of servicing, being a real partner with our clients, we spend an awful lot of time and effort making sure that we are providing not just good products and equipment, but good education and content, making sure that you're available or you're aware of what is coming to the ornamental landscape near you, so to speak. So that's why, especially with beech leaf disease, we've spent a lot of time, working with cooperators, really figuring out what's going on and what we can do to manage this significant issue. So as I mentioned, kind of our agenda for today is talk a little bit about beech leaf disease, where it is, kind of where it may have came from, get into the biology of it, and then with that kind of context in mind, getting into the management and some of the research that's going on in respect to that management. So beech leaf disease was first detected in Lake County, Northwest Ohio back in 2012. And it didn't really move far very quickly from there. It kind of stayed, that map to your right is as of 2020. And you can see it's kind of stayed in that pocket of Northwest Ohio, Northeast Ohio, Northwest Pennsylvania, and kind of stuck there. The nematode is suspected to have come from overseas, and it is most closely related to a nematode also called Littlelinchus crenatiae, which we'll talk about in a little bit. But that was a nematode that was most recently, discovered in 2019 on Japanese beech. So that was also sort of a new nematode discovery. And so that's where we think that there's a possibility that it came from there. But it's really, I don't think there's anybody that's really quite sure exactly where it has come from. That being said, since being introduced into the United States, it has spread out to quite a large area. You can see here that it's made its way all the way up into Maine. It's up into Ontario now, and it's as far south as Virginia. There's also been some detections as far west as Michigan. So it has certainly over the last 4 or 5 years um, spread out quite a bit more than it has the first few years of it being here. That could be attributed to its actual spread, or it could also be attributed to more folks out there actually looking for it. So it may have just not been detected, but been in some of these areas prior to being officially detected, if that makes sense. So take this with a grain of salt. But nonetheless, if you're in the Northeast, this is certainly something that you're probably seen, or if you haven't seen it yet, you probably will see it. So definitely be on the lookout. And this is really also only just a portion of the native range of our American beech. So we really, fortunately or unfortunately, really just scratched the surface of where this nematode potential range could be. So we may see this map continue to increase as the years go on. But what exactly is beech leaf disease? You've heard me kind of say nematode and beech leaf disease. What are we actually dealing with here? Well, beech leaf disease is caused by or associated with a foliar nematode, Litholinchus crenatiae mccannii. For your sanity and mine, I'm going to refer to it as LCM from here on out. But that's what it's most similar to. As I mentioned earlier, it is similar to a Litholinchus species that was first described back in 2019 that is also associated with leaf gall symptoms on Japanese beech. However, it is important to note that that nematode does differ slightly in morphology and some DNA markers, which is why we have the mccannii tacked on to the end of there. It is slightly different. The other thing that's really interesting with this nematode is you might be thinking to yourself, well, how can a microscopic nematode, because that's what these are, they are microscopic. You do need electronic magnification in order to see these things. You're not going to see them with the naked eye. So how can how can something so small cause so much damage? And one of the reasons for that, we'll talk more about it in a bit, is that there are thousands and thousands and thousands of these things that are being found in bud tissue and leaf tissue. So you have very high populations of these things, and their feeding then leads to that gall or banding symptoms that we are probably already familiar with. But we'll talk about it if you're not. As far as the life cycle, and this is something that's really important to keep in mind as we get further down the line into management, because we have to make sure, just like with most of our other pests, that we are utilizing our management techniques at the correct time in order to get the most effective control. And so, interestingly enough, right now, as we are sitting here talking about it, the nematodes are in the bud actively feeding. They feed in the bud all through winter. And as the leaves break bud, they are then dispersed, and those eggs are dispersed throughout the canopy. And then throughout the summer, we don't see much activity from adult nematodes. However, as we get into late summer or mid to late summer, July, August timeframe, we see adults starting to colonize bud tissue. And then that's where they'll be for the remainder of the winter, feeding, and the cycle then repeats. So, a few important things there that we'll talk about in a bit, but this is super important when it comes to our management, because once again, we don't see much activity during the summer, and that's a key piece of the puzzle here. Now, how are these nematodes getting to asymptomatic beech trees, right? How is it spreading? Well, for quite a while, there was a lot of speculation. We were speculating that, obviously wind and rain splash and lots of biotic factors. One thing that I've said before, and I think is still something that's quite plausible, is that there are bird spread. And fortunately, there was some work done by Penn State and the United States Forest Service that was looking at this specifically. And this study was just published late last year, and it did kind of confirm some of our speculation. So it's always nice to know that what you were kind of speculating was actually true. And what was interesting is that wind and humidity was found to be, as far as local spread goes, a big factor, as you may have kindly guessed. Interestingly enough, what they also did was they took live caterpillars from symptomatic beech foliage and inspected the frass and were actually able to find live LCM in the frass of caterpillars, meaning that there are some additional biotic vectors, if you will, that are out there. Some other interesting things that require more study is if there are birds feeding on these caterpillars, and this needs more study, but it is something I found a little bit interesting, is are these LCM able to move through the digestive tract of a bird and still remain viable out the other end, so to speak. And that's something that still does need some more research to evaluate, but interesting nonetheless, and something to think about. So still some, more work when it comes to vectors. I think especially our biotic vectors, but nonetheless, as far as the local spread goes, this is some really great work and some really good research that's happening out there, and the first of its kind, quite frankly. Everything up until this point's been a lot of just speculation. So, as I mentioned before, the nematodes are feeding right now. And as a result of this feeding, we start to see the symptoms that we're probably very familiar with if we live in an area where beech leaf disease already is. And that is this dark interveinal banding that shows up. On our American beech, it's really easy to spot, especially if you're looking up into a canopy on a bright sunny day. It's very easy to see the contrast between the dark banded part of the leaf and the normal part of the leaf. So it's very easy to spot, and it's a very characteristic symptom of beech leaf disease. There aren't really any other biotic or abiotic issues that are going to cause this type of symptom pattern in beech leaves. And so it's a good diagnostic clue. And like I said, it's very easy to spot. So keep an eye out if it's not something you've seen yet or noticed yet. Certainly be keeping an eye out, especially on those American beech. It's the easiest to see. And that banding also does show up on our European copper purple-leaved varieties as well. It's just not as easy to notice. I think it's also important to understand that, we talked about Americans, we're talking about coppers or Europeans. It's important to note as well that as of right now, there is not a resistant or immune species or cultivar of beech. As far as I'm aware, there are, I've seen it on fern leaves and weeping and copper and just about any of them, the tricolors, you name it. I've seen that banding on pretty much every species and cultivar of beech that I've run across so far. Maybe in the future, there will be certain species or cultivars of beech that are identified as resistant, but for right now, if you have a Fagus species, I would certainly be on the lookout for this issue. Now, as I mentioned, with our copper beeches and purple leaf varieties, that banding can be tricky to spot. It is still there, however, because of the dark leaf color already, it's a little trickier to notice. But what you normally wind up seeing is sort of the second stage of damage, if you will, and this is usually as a result of defoliation within the season, and then refoliation. And when these new leaves come out mid-season or so, they are very tattered. They look almost herbicide damaged. They can have these large kind of like blistered appearance, and they just look raggedy and tattered and terrible. And it's important to note here as well that symptoms don't progress throughout the year. In other words, the banding is not going to get worse as we move through the summer. The banding is a result of the feeding that's occurred in the winter. And therefore, if you're scouting for this and you inspect a beech tree in late May after after the leaves have fully expanded and you're not seeing banding, well, then you're good to go there. You don't have to continue to come back to that tree later in the year to continue to scout. If you're not seeing symptoms in the spring, you're not going to see symptoms later on that year. So that's kind of a nice thing for it. However, there's always exceptions to those rules. And what we have come across a few times is asymptomatic leaf tissue testing positive or having confirmed nematodes in them. They just haven't quite reached a damaging population yet. So you certainly, probably don't need to sample every beech tree in your area. I think it is safe to wait for that banding and then react to it. But if you have specific questions about that, we can talk more about that offline. Now, as these symptoms get worse, and usually you're going to see these kinds of symptoms, leaf symptoms, after your canopy is already pretty thin. But the leaves that are remaining do tend to get sort of this leathery, very thick texture and feel to them. And once again, this is usually after this is usually a couple years after initial detection. And so the disease is it's part of that natural cycle there, if you will. So speaking of thin canopies, that's kind of the later stage. As this disease progresses, we usually see thinner and thinner canopies year over year over year. In these three examples here, these are all about 4 to 5 years after initial detection. And you can see, especially those pictures on the left and right, very, very thin canopies there. And we can see mortality sometimes in as few as just 3 to 4 years. However, in most cases, we don't see mortality, especially in mature trees, until 5, 6 or so years after initial detection. That's not to say that's going to be the case across the board. There are some extreme examples that I've run into or heard of asymptomatic trees, becoming symptomatic and then within 2 years not leaving back out. So there certainly are exceptions to those rules. However, in most cases, we're seeing trees hang on for at least 5, 6 years before really becoming pretty poor health and then ultimately dying. So something to keep in mind there. Unfortunately, for those of you who are on the eastern portion of this disease's range, it does seem to be progress a little bit faster and be a little bit more severe. That could be because of some of the other issues going on. For example, we're dealing with beech bark disease and things of that nature, Phytophthora, things like that. That could be part of the reason that we're seeing faster mortality occur, or at least faster progression occur on the eastern portion of this disease's range, but just something to keep in mind there. And what we're seeing is, as a result, the damage, if you will, is quite interesting. So we all know that trees have a carbon budget, and this doesn't matter if we're talking about beech or maple or whatever, right? Trees have a carbon budget. They have they're photosynthesizing to create these carbohydrates. The energy is distributed to various functions within the tree, various organs, and some of that energy is stored and used for the following growing season. Now, the reason I'm saying that is because in beech, there are a few things that can occur that can have a drastic impact on resource allocation or just resources that are available. For example, when beech have a heavy fruit set, the following year we can see as much of as a 20% reduction in growth wing width. I'm going to stop trying that one. The following year, so a significant reduction in growth the following year. In addition to, and this is something that's really interesting that I found out through a book, it's Applied Tree Biology, is that 10 to 20% of the wood in an annual ring, when it comes to Fagus sylvatica, is funded by the carbon reserves of the previous year. So that's really interesting, and it's something that kind of helps add a little bit of context as to why we're seeing such quick potential mortality from this disease, and that is it's sort of this domino effect that's occurring. So you have already the tree is able to be is less efficient because it's either losing leaves throughout the season, or the leaves that are out have this banding, making them less efficient and less able to make the food required, the energy required, which means they're going to be able to store less for use the following year. In addition to that, and this is some work that was done from the Holden Arboretum, and I found this very interesting as well, is that below ground effects could also be significant, especially when it comes to mycorrhizal associations. So mycorrhizae, they have this symbiotic, this mutually beneficial relationship to the roots that they colonize. And so what this paper was looking at is that relationship affected when trees are suffering from beech leaf disease? And the answer was yes. It could have, in some cases, a very significant effect on the ectomycorrhizal populations that are colonizing those root tips, making the roots less able to pull up the nutrients required for tree growth. So you have sort of this 1-2 punch there. Very, very interesting. And something else to kind of keep in mind when it comes to this. Now, when it comes to the biology, I'm going to kind of stop there. I could spend a lot more time on that, of course. However, if you are interested in kind of exploring more about this, there's a ton of research. There's a ton of papers out there. This is one that I like referencing to because I think it's really well written, and it breaks down a lot of the facts of why beaches are so important to our ecosystems, the effects of what's going on with beech leaf disease, as well as breaking down a little bit more of the biology, if you will. So something to reference if you're interested in more, but there's plenty of documents out there. And this is where I want to start to get into the management, because this is really, let's say, where the exciting stuff is, right? Because unfortunately, for the last, since 2012, really up until about 2023, we really had no good answers as to what we could do to manage this disease and preserve the health of our beech trees. And so we do have answers now. We do have great management options that are available, and that's what I want to spend some time talking about. But first, I want to kind of address some of the things that have not worked. The products that we've looked at, and when I say we, I mean a large group of researchers that have been looking at this, not just us here at Rainbow. But what we have looked at so far is products like emamectin benzoate, abamectin, acephate, among many others that unfortunately just did not work. Now, a lot of these products, they did work in a lab environment. In other words, emamectin benzoate is a very effective nematicide. However, the problem is these products did not get to where we needed them to get to in the tree. Remember, the feeding is occurring, the damage is occurring in the bud tissue. So we had to find, or have to find, a product that's going to move into the bud tissue. And unfortunately, the reality with many of our systemic products that we tried is that they did not make it in high enough concentrations to contribute to any management into that bud tissue, and therefore were ineffective. On the flip side, though, just about 2 or so years ago, Bartlett was doing some work with fluopyram. And what they discovered is that Broadform, which is and it's an NVU product, is very effective against the nematodes. It's a foliar applied product, and when applied with the proper timing, is very effective at managing nematode populations. In addition to that, over the last few years, we have been working with Bartlett to study thiabendazole. Some of you may be familiar with this, maybe used it for Dutch elm disease in the past, or sycamore anthracnose. And so that is also another product that we have gotten great results on that we'll dive into today as well, which is really exciting. And then the first one we're going to talk about here is phosphites. So there have been a few people that are out there working with phosphites, working on phosphites. Davey has a few study results published and are continuing to work on phosphite applications. So we'll talk about that first. So let's talk about potassium phosphate applications, right? We know that that is an extremely valuable tool for us across the board when it comes to plant healthcare. It's a tool that's used quite widely and has quite a number of different use cases. Um, the really nice thing about potassium phosphate is that we can apply it a number of different ways, right? We can do soil drenches with it. We can do bark sprays. We could inject it if we really, really wanted to. There's a number of ways we can apply it, and it's very systemic. It moves very well throughout the vascular system of plants. It's also very effective at managing certain diseases, Phytophthora species being one of the main ones that it's used for. But it also does a lot of other things that are beneficial. For example, increasing lignification and the thickness of cell walls, as well as enhancing the production of plant defense compounds. Um, so it has a lot of really good benefits when it comes to just overall kind of tree health and stress amelioration and things of that nature. And quite frankly, there are some studies that are ongoing that are looking at phosphate applications, and some of these studies are seeing a reduction in nematode populations as a result of phosphate applications. Now, I think the jury is still out as to why we're seeing those why we're seeing those nematode populations reduced. However, it is something interesting and something that we can look forward to seeing possibly research work on in the future. As of right now, in general, we're using phosphates to enhance the defense mechanisms of the treated hosts, right? Rather than using them to directly manage diseases. And that's still kind of, I think, where my position is on this based on what I've seen. However, like I said, there is work ongoing with this, so that position may change. For now, though, I think the recommendation that I would give when asked is using phosphates in conjunction with one of the other two products that we're going to be talking about, simply based on what is published and available right now. So just something to keep in mind there. If you are interested in using phosphates as part of the protocol for managing beech leaf disease, there are a number of different trade names out there. Doggett carries Dogfight. Plant Food Company carries polyphosphate 30. There's many of them out there. They're easy to get your hands on. And as far as rates go, this is specific to Dogfight, but the rates are easily found on the label. Generally, this is an application you'll be doing either in spring or late summer, or if interested, you could also do them both times as well. If you're interested in learning a little bit more about some of the other products that didn't work, maybe you're just curious, as well as getting a little bit more context and information on fluopyram, which we're going to cover next. This is a research paper that was published about a year or so ago that looks at a number of different active ingredients, including fluopyram and oxamil, both of which were effective. We're not going to talk about oxamil because the reality is that it's probably never going to be labeled for our uses in the ornamental landscape. And at this point, we have better systemic products anyways. However, fluopyram is a product that may be your choice to go with when it comes to management. So fluopyram, or broadform, that's the trade name for it. It's a fungicide and a nematicide that was approved for use back in 2012. In essence, it paralyzes the nematodes, and it is extremely effective against LCM. It is very, very, very effective against the nematodes that are responsible for beech leaf disease. I've said in the past that resistance might be a concern. However, I have since learned more and realized that that is not actually the case. For one thing, this product is very effective against the nematode. In addition to, we don't have a very wide genetic diversity when it comes to this nematode. So therefore, it's not as likely that we're going to see that resistance. Additionally, and we'll talk about this in a bit, is there has been additional work done by Bartlett that has looked at reducing the rates of fluopyram. So we were using the highest labeled rate, which I believe was 8 ounces per 100 gallons. And since that research work, that has not only prompted a new version of the 2EE label, which allows for lower rates, but also has paved the way for recommending lower rates in general. So we're able to now use half of that and use a 4 ounce per 100 gallon rate, which is really good. Makes it more cost effective, as well as lower potential impacts on the surrounding ecosystem. So something to keep in mind there. Now, when it comes to fluopyram, this is a foliar spray product. We have to apply this as a foliar spray. So right away, some of you might be thinking, I don't want to do that. I am sick to death of spraying things. If I don't have to spray it, I don't want to. However, keep in mind that for our smaller, especially ornamental, like our weeping beech and stuff, this might be the preferred option. And once again, there is ongoing research work determining the optimal timing. I think for sure you're going to want to have at least one application that you're doing late in the summer to control any nematodes that are moving from untreated beech onto treated beech, beeches that we're looking to manage this on. However, there is some very interesting work that is not quite published yet, but in the works, looking at additional timings for these treatments. I'm not going to get into that today. If you're interested and want to chat more, we can chat offline about that. But something to keep in mind there. The key thing with fluopyram treatments is that we are spraying and getting this product onto the nematodes prior to them colonizing the buds. Once these nematodes have made it into the buds, these sprays are ineffective. This spray does not get into the bud tissue. So we have to get the nematodes prior to them colonizing the buds. That is the key. The other thing that's important is we have to get thorough spray coverage. And when you're talking about these large mature beech trees, that can be a daunting task. And so making sure we're getting full and complete spray coverage is important to making sure that we're getting that predictable management. As I mentioned, we do have lower rates now that are available in the States with the updated 2EE label. I went ahead and just pulled a copy of this to show you. And you can see the previous version of this label did specify 8 fluid ounces per 100 gallons. The new 2EE label gives you a range now, so you can lower it to 4 ounces. And based on research, once again, that Bartlett Labs has done, that 4 ounce rate is just as effective as the 8 ounce rate. So you can, and I would go as far to say you should be using that lower rate this year and into the future. So maybe you're thinking to yourself, well, that's all really great and exciting, but maybe I can't spray because I have a waterway or other things, or it's just too large. I don't have the equipment. Well, the good news is that there is now a systemic management option. Now, we've been talking about this at this point for probably about a little under a year. And this is really what, to me, is exciting. Obviously, we'll address the elephant in the room. Arbortect is a rainbow product. However, what's really exciting about it, and what should be exciting to you, is that it's a systemic product, meaning that we are not required to get full spray coverage. We're not even spraying this stuff. We're injecting it. So the active ingredient in Arbitect 20S is thiabendazole. Historically, thiabendazole has been used in veterinary medicine as a dewormer for cattle. It was used for a time even in human medicine. However, it has since been replaced by much better medications. So if you go to the doctor and he's trying to prescribe you thiabendazole, you probably just want to find a new doctor. But the reason that it's effective for against Beech Leaf Disease is that it is an anthelmintic, meaning that it is effective at preventing worms from growing and or multiplying. And that right there is the key. The other key is that it does get into the bud tissue. It also moves into new wood, which is why we're able to see multiple years of control from one application. Now, I'm getting ahead of myself a little bit because I get excited talking about this, but it's a systemic product. And we'll talk more about what that means in a minute if you're not familiar. So We apply it via what's called a macro root flare injection. And basically, if you're familiar with maybe micro injections, for example, if you've done emerald ash borer treatments with emamectin benzoate in the past, right? That is a micro injection where we're drilling a hole at the root flare of the plant, of the tree. We are injecting a few milliliters of product in, and it's moving through the vascular system that way. The difference between micro and macro, as the name suggests, with macro, we are just using larger volumes of material, of solution. And so instead of talking in terms of milliliters, when it comes to macro injections, we're talking in terms of ounces and gallons. So we're just putting more solution, more product into the tree. Otherwise, the basic principles still apply. And we'll talk about, we'll get into the weeds with that a little bit as we move on. As I mentioned too, this is effective for multiple years. So one application is effective for up to 2 years. And that's really great because if you haven't picked up on it yet, this does require some mechanical wounding of the tree. We are drilling, albeit small injection sites, we are drilling small holes into the root flare of these trees in order to carry out these types of applications. So not only do we have to, not only are we able to now wait longer between, making that damage and doing these injections, but it's also generally going to be more cost effective and more efficient to do these types of treatments, not only for your clients, but also for your kind of the way your business works. Because once again, it's one visit that's going to last 2 years. So if we had applied in the fall of last year, we would expect to see a reduction in symptoms and damage this year as well as next year. And we would reapply at the end, or we would reapply next year. The other thing that's important too is that Arbitect 20S is labeled, it has a very specific label. A lot of our labels and a lot of the products we use has a rather broad use range to them. In the case of Arbitect, it's a label that's written kind of odd. In other words, it's only labeled for Dutch elm disease and sycamore anthracnose, and any other use of it is off label. And so what we had to get and what we have gotten now are these 24C special local need labels, and we have them available in select states. And we'll talk a little bit more about that in a minute. But first, I want to show you some of the data. So as I mentioned, we're very fortunate to be able to partner with Bartlett Tree Research Labs on this work. And this is what we found. So this is our 2-year data. And what you're seeing here is in the blue, that is 2022, and the red is 2023, and the green bar is 2024. And in the group to your left is our Arbitect treated trees. And in the group, let me see if I can make this laser pointer work here. So our group here to the left, these are our trees that were treated with Arbitect. And then on our right, this group was our untreated controls. And what we're looking at here is the percentage of canopy with symptomatic leaf tissue in it. And what you can see here is that the 2 years following, so remember, this is the year that we applied, and the year we apply, we don't expect to see any management that year. So this is basically our initial ratings. The following 2 years, we had a dramatic reduction in leaf symptoms. And that right there, that's the key. Versus our untreated controls, where you can see there was still a slight reduction in leaf symptoms, and that could be chalked up to a number of different reasons. But you can see our untreated symptoms versus our treated symptoms, still a very significant reduction there. The other thing that we looked for, and we were a little bit maybe concerned about, I guess, at first, was, are we going to see any phytotoxicity? And throughout all of the trials, there's about, I'd say about half a dozen of them or so scattered between Ohio, New Jersey, and Connecticut, and Pennsylvania. And we haven't observed any phytotoxicity as a result of these injections thus far. So that's also a really positive sign. The other thing that we've seen, and this is really, I think, almost more important than a reduction in symptoms, is we've seen essentially no increase in dieback the years following application of Arbitect. So once again, 2022 in blue, 23 in red, and 24 in green. 22 is when we did these applications, and this was how much dieback was observed. And you can see it looks like there's a slight reduction. That's just due to the way we rated these, but basically there was no increase in dieback the following 2 years. We were able to stop that basically in its tracks. That is a big deal. And when you compare that to our untreated group, you can see that we did have an increase in dieback the following 2 years after after we were out there applying. So untreated, we did still see a continuation of that dieback. We did see increased dieback. However, on the trees that were treated, we did not see any increase in dieback at all. And that is really, really, really exciting. Now, if data is not your thing and pictures are cool, here's a picture that's a great representation. Now, keep in mind, this is 11 months after treatment. So these applications were done in late August of 2023, and this is July of 2024 when we were out here. So this is only 11 months after treatment, but this is a great representation of what you should expect to see as a result of Arbortect treatment. So on our left here, this beech tree is left untreated, and this is our treated tree. You can see much fuller canopy, much healthier looking canopy. A significant difference there. And just also to give you a little more context, this is pretty much what all the canopy looked like when we were out there doing our initial applications and ratings. So this is a woodland, residential woodland, and they were nice enough to let us on that property. We did close to, I think, 40 to 50 trees out there. And so this is just a really nice representation of what you should expect to see years following Arbortect treatments. Now, as I mentioned, 24C labeling is important. I inadvertently put Michigan on here. Michigan is not approved yet, so just ignore that for now. However, we do have 24C labels approved in the rest of these states, with New Hampshire being the most recent one. All of these labels, I believe, can be found on our website underneath the Arbortect product page. So if you are using Arbortect underneath the 24C label, if you're using Arbortect for beeches, make sure you have a copy of that 24C. It's a supplemental label with you. Just make sure it's available with you. But these are all available and can be found on our website. Now, the other reason that multiple years of efficacy was really exciting is that because one of the stipulations in this 24C label was that we cannot apply this product any more than once every other year. So not only are we able to follow the label, but we're also going to get really effective results as a result of one application. So really exciting stuff there. Let's talk briefly about the macro injection process, and I do want to leave a few minutes at the end for questions. And if you are able to stay longer, Kendra and I can hang out for maybe an extra 5 or so minutes after the hour to address questions. But I want to talk about the macro injection process because there are some nuances to it when it comes to injecting beech trees. Some of you might be thinking to yourselves, "Well, I really don't want to inject beech trees," and that's because we know them as sort of sometimes slower to compartmentalize, slower to form that reaction or response growth. However, some really positive things when it's come to injecting beeches, but also some things to be aware of. So really quickly, for those of you unfamiliar with the macro injection process, basically what we have here is this bucket here is our reservoir. The bucket is, and this is our pump. And then what we have is basically just a harness system that has a bunch of these orange injection tees. And what we're doing is we're pumping the solution out of our reservoir through our pump and into our harness here, which then the tree is able to take and pull up via its vascular system. So pretty simple in theory and also very effective. We've been using this method to inject trees for close to 5 decades. So this is not a new procedure. It's just a new use for an existing procedure. So we've been injecting trees forever. This is just a new use case for it. If you don't have a system already, fair warning, this is a little bit like IKEA, where you have to kind of build it yourself. This is what you get. So you just get a big roll of tubing, a bunch of these orange tees, and you do get to put it together yourself. It's kind of fun the first time, but something to keep in mind. So if you are going to order a system in anticipation of doing these treatments, make sure you give yourself an extra day or two to put it together. As far as our rates go, we're going to use an equivalent rate to our sycamore anthracnose rate, which is kind of our mid rate, if you will. So we're not using as high of a rate as you would for elm trees. So it basically comes out to 1.6 ounces of Arbortect, which we dilute into 51.2 fluid ounces of water, and we do that for every diameter inch. So we do have a really nice chart. This one is actually out of date a little bit, but we do have a nice chart available that you can reference if you do a lot of these treatments. It makes it a little bit quicker and easier, just less math. When it comes to Arbortect, I'm going to breeze through. When it comes to Arbortect, water pH and water quality is very important. Once again, if you're interested in finding out a little bit more about this, we can talk more offline. But water quality is very important. Make sure you're using neutral to slightly acidic water source, as well as a very clean and fresh water. You don't want to be using, water out of a creek or, anything like that. You want a very clean, as well as neutral to slightly acidic water source. Otherwise, you can run into some problems with Arbortect mixing in well. Then, as I mentioned here, so one of the things that we found when we started injecting all these beech trees is that we did see some cracking and fluxing of some of the injection sites when we went back to do evaluations. And that was a little bit concerning at first because, for one thing, it just doesn't look good. Now, those injection sites were still sealing over nicely. It just didn't look good. And what we found is that when we started to take our injection sites, and rather than injecting on the tops of these flares, you can see how I kind of avoided the top of the flare here. And in focusing our injection sites on the sides of the flare, we saw a dramatic reduction in how many injection sites had cracking or oozing, fluxing after injection. So we were able to reduce dramatically to, it was almost, we didn't really hardly see it very much at all after moving our injection sites to the sides of the flares. So that's one of those nuances that I talked about. If you're going to be injecting beech trees, it would be in your best interest to, instead of injecting right on the tops of these flares, just simply moving over to the sides of the flares, just so that for whatever reason we saw less of that cracking. And I don't want it to scare you. It wasn't super dramatic. It wasn't, as far as the overall health of the plant goes, it wasn't a big concern, but it definitely didn't look that great either. As far as your timing for these applications go, we can do these applications anytime after full leaf expansion, all the way up through, depending on your area, September, early September, I would say. As you get further and further north, you want to get them finished up probably closer to late August. However, your ideal timing is definitely going to be between full leaf expansion and July. You really want to get, ideally, if possible, you want to get these injections done prior to the end of July. And the main reason for that is because, remember, these nematodes are starting to move into the bud tissue in that kind of mid to late summer time frame. So if we're waiting until August or September to get these treatments applied, we're going to see improvement the following year, but we might see, as a result of the feeding that's occurred between them getting into the bud and our treatments occurring, we might see some banding the following year. So keep that in mind. The later you wait to do these applications, the more likely it is that you see banding the following year. But the good news is that you will still see an overall improvement in the health of the canopy, as well as you should see a reduction in dieback as well. So ideally, get these treatments, these injections done prior to the end of July. However, if you're getting calls at the end of the season, don't turn them down. You still want to get the treatment into the tree as soon as possible. So, but it is important there to kind of set the expectation that you might still see some symptoms in the canopy the following year, only because we're getting on this treatment a little bit later than what's ideal. In addition, and this is definitely more anecdotal, this is something that I've experienced as I've been doing these injections over the last few years, is that when you have a heavily symptomatic tree, in other words, tree where the canopy is showing 60% or more symptoms, it might be in your best interest to wait until later in the season, say June, to do those treatments. What we found is that by waiting sort of for that kind of second flush of leaves, that we get better, more efficient uptake, or our uptake's a little bit faster. When we treat those heavily symptomatic trees in early May, we were seeing just longer uptake time. So once again, it's just something to keep in mind. That's something I've seen. It may not be the same across the board, but I figured I'd throw it out there just in case any of you have experienced that same thing. As I mentioned, I want to leave some time here, so I'll talk about this quickly. But you know, when it comes to macro injections across the board, you really want to give yourself every possible opportunity for success, meaning that it's the little things that make a big difference. So using a sharp, clean, in good condition drill bit is super important. Really following those best management practices when it comes to tree injection, which we just don't have the time to talk about today, but following those best management practices, evenly distributing your injection sites, making sure that the teas are inserted correctly. All these little things really can make a big difference when it comes to how efficient or how well that product is going to be taken up. When it comes to micro injection, a lot of times we can get away with, this, that, or the other thing. When it comes to macro injection, though, we're talking about, once again, gallons of product or gallons of solution that's being, delivered to this tree and that we're expecting this tree to take up. So those little things can make a big difference. So spend the time to set up your injections the right way. Wait for those optimal conditions, decent soil moisture, fair weather, that sort of thing. It will go a long ways in really making you successful at these applications. As far as your uptake timing, we're getting here to, we're wrapping up here. As far as uptake timing goes, under optimal conditions. So once again, that's when, the weather is decent for injections. You have good soil moisture, not too wet, not too dry. And then you're doing everything on your end properly. You should expect the uptake time to be somewhere in the neighborhood of half hour to 45 minutes, sometimes up to an hour, depending on the size of the tree, of course. So what that means is expect to spend anywhere between an hour to 2 hours, an hour to an hour and a half on site to give yourself time to set up, and then give the tree time to take up. As you get into larger trees, you may want to increase that time accordingly. And then once again, in those early applications of highly symptomatic trees, you can see those trees sometimes take up to 2 hours. So that is one of those caveats and things to keep in mind. So as we wrap up here, and we have a few minutes for questions. As I mentioned, this is an evolving protocol. What I say today very well could change, hopefully not tomorrow, but in the future as more research comes out. There are still a lot of unanswered questions when it comes to beech leaf disease. There's still an awful lot of research being done across the board when it comes to beech leaf disease. So this is something that you're going to hear. Well, you're certainly going to hear us talk about it quite often. It's going to be a hot topic for probably a very long time. And if you're interested in keeping up, we do make every effort to update our own information, which you can find via our website, when it comes to especially management recommendations, as well as we'll continue to do these webinars periodically, maybe even bring in some other speakers if you're sick and tired of hearing me talk about it. That's fair. So we're going to continue to update you and keep you up to date on what's going on when it comes to beech leaf disease. And with that, if there are questions that we don't get to within the next 5 to 8 minutes or so, this is my contact information. I'd love to hear from you. So if we don't get to you online here, feel free to reach out offline. So, Kendra, do we have any questions?
Awesome. Yes, sir, we do. Thank you so much. That was a great webinar. So we had a number of questions that I was able to answer live, but we've got a few left here in the Q&A box. So we're going to go back to the very beginning. And Jay Webster says, whoa, hold on. The nematodes were found live in the frass of caterpillars. Is it possible that they could be present in a moth or butterfly?
Ooh, yeah, it's possible. It's plausible. I would have to. What I'll do is I'll share that research work with you. It is open source, so I can share that paper with you. Um, I would review that. And I don't know if they addressed that specifically in the paper or not, but that would be where you would find that information for sure, one way or another.
Mhm. Thank you. The next one is from Steve, and Steve says, is there any hope or reason to believe a natural predator or parasite may emerge for the nematode as their populations increase?
Yeah, I haven't heard much talk about that yet. That might be partially because most of my focus has been on what can we do to manage it. That's certainly taken up the majority of my bandwidth, if you will. However, I'm sure there are folks looking at it. I know there are folks that are out there that are looking at what's going to replace the beach, especially in forested ecosystems. Um, so, as of right now, there's always reason to hope, but I don't think there's any. I haven't heard of any kind of specific work being done on that, or if there's been any progress with that work. So as of right now, I would say there no.
Okay, thank you. And just to add a little bit to that, Steve, there's always hope that there's something either native or non-native present here in North America that could step up and help to control this nematode. I believe current research shows that the nematode is originally from Japan, or at least Japan is its native range. And so there's potentially a natural enemy present in Japan that we could explore introducing here. So
Yeah.
absolutely still more research to come on that.
Yeah, and the other thing that I found really interesting is that the closest nematode that was found, or the most closely related nematode that was found, was only identified or named in 2019. So, I mean, this is really new. It seems like almost across the board. So, like I said, a whole lot left to learn here.
Absolutely. And Mark and I are going to hang on for just a few more minutes. I do realize we're at time, so I want to be just respectful of people's time today. For those of you that have to hop off, just thank you so much for joining today. And please don't hesitate to reach out if you have any further questions. And a recording of this webinar will be available on our YouTube page as well.
Yes,
So getting,
and if
go ahead.
sorry, one more thing on that, Kendra. Give it about a few weeks for this to be reflected on your ISA statement. So if you're looking for credits, there's nothing you have to do. We're gonna submit this on all of your behalfs, but give it a few weeks to show up on your ISA statement. It does take some time for it to be processed.
Yeah, absolutely. Okay, back to questions. So Jay says, so caterpillars that balloon could be spreading the nematode.
Yes. Yep. That was an
Possibility,
easy
yeah.
one.
Yeah. Okay. Would you recommend treating preemptively if symptoms are not present?
No, I would not. I think that's where the phosphates would play a very important role, as well as some of the other cultural other cultural things that we should be doing. So, if you're in an area that maybe it's been detected in your area, but you're not actually seeing those symptoms, I would not recommend you treating at this point. There's just no need to. However, take the soil sample, mulch that tree if you can, do all those really great cultural things ahead of time. That is absolutely would be worth your while to do. But I think as far as actual, Arbitect or fluopyram treatments, I would wait until you're seeing symptoms.
Awesome. Thank you. Next question. What is the point of no return for trees with percent canopy decline? Once you see presentation, is it too late or is there time to treat?
So I would say I've seen trees that 80+% of the canopy is showing symptoms and still treated them with success. The key differentiation here, and you kind of touched on it, is decline or dieback. And what I would do is I would say pretty much the same kind of parameters we use for other injections. So if you're seeing, 30 to 40% of dieback, which is different than symptoms, then that's where I would probably, at least have a conversation with the client, or just not treat at that point. But that's where I would kind of start to really think about, is treatment going to be worth it? Is that 30 to 40% dieback is kind of that, where I would say it. If you're seeing any more than 50%, I wouldn't even bother. I think it wouldn't be worth it. So, yeah, hopefully that answers the question.
Absolutely. Thank you. Next question. Would any of the working products to control beech nematodes work on pine nematodes?
Um, well, Arbitec, that's the easy one. I would say even if it does, we can't do it anyway. So I wouldn't do that. As far as fluopyram, I'm not sure. I just don't know the answer to that.
Okay, thank you. And in a similar vein, what's the best timing for fluopyram and phosphite injection applications? Sorry.
Yeah, so I knew that was a question that was coming. So for fluopyram, and that's why I mentioned it earlier, but I think there's right now the research that is published, those applications were done starting in mid to late summer and continued on through fall. So you were doing basically what that research, when they were doing in that research, is they were starting their applications, I believe it was in late August, mid to late August, and continuing in every 3 weeks, basically through September. And so that is what published data is out there. As I mentioned, there will be more data published probably sometime this year that's looking at alternative timings of those applications. But for now, I would simply that's the timings I would use for now. Until that research is published and available, I would stick with the late season multiple applications. We know it works. We have data to back it up. For phosphites, spring or fall, or both.
Okay, thank you. That actually answers the next question too. Jessica says, "Hi, Mark. Will you touch on the combination of treatments or the recommendation for trees that were injected with Arbitec last season? Is the recommendation for foliar broadform applications in the bi-year?"
So no, you're going to use one or the other. If you have injected with Arbitect, then you don't need to spray the following 2 years. The Arbitect by itself is effective enough where you don't need to supplement that with anything else. And the same goes for fluopyram. If you're doing a fluopyram spray regimen, there's no need to inject. So what I would say is pick one or the other based on the situation you're in and stick with that, at least within the season.
Okay, thank you. Will 2 ounces per 100 gallons of fluopyram work?
Um, yes, but it's off-label. That 2EE has a specific range you can use. So I would stick with the 4-ounce rate if it were me.
Okay, thank you. These products are allowed to be used in residential areas.
Yes. Now there are certain restrictions on the label, so review the label of whatever product you're using to be aware of them. For example, both are highly toxic to aquatic invertebrates, so you do have a buffer zone there, but that's all on the label. So review the label, but yes.
Okay, thank you. And I'm going to skip down to some of these bigger questions. When it comes to water used for both micro and macro injection, is city tap water okay, or should we use distilled water? My concerns are impurities, chemicals, and metals in city water and negative effects to trees.
Yeah, so the key thing, that's a great question. I touched on it briefly, but the key thing with your water source is that it is city water is absolutely okay. The only issue that I've run into is a lot of times city water is a little bit too, excuse me, it's a little too alkaline. So we would have to use some sort of pH buffer. Muriatic acid is the one that's most that I would recommend you use, or running that water through a deionizer filter or deionizer pump. Either one of those options would work. That would help to filter out some of the impurities, as well as bring it to, like I said, a neutral to slightly acidic pH level.
Okay, thank you. Will there be a 24C label for Maryland?
Maybe. It's pending right now, so we're just waiting on approval. So hopefully soon, but I can't say for sure.
Okay, perfect. You mentioned the spray application of flupyram may have reduced efficacy when near other infected beech trees. The thiobendazole injection treatment was right next to the OTC tree. Is the systemic thiobendazole treatment more resistant to reinfection from nearby untreated trees?
Yes, yeah, and that's really one of the key advantages of it is that even if you're treating a tree that's surrounded by untreated beech, because that product is staying in that tissue, that's why we see continued control for that full 2 years. So if you're only treating one tree in the midst of a bunch of untreated trees, that's definitely another reason to go the Arbortect route.
Okay, thank you. Just looking through these questions here. Can you inject Arbitec 20S with different injection equipment? I currently use an injection tool from a competitor which injects one site at a time.
To be honest, no. It would take you forever. And, once again, understanding that I am coming from Rainbow, obviously, I will say that the injection equipment you want to use, it is very cost effective. It's not expensive. It's durable. It's good equipment. It's not gonna, you're not gonna have to mortgage your house to buy it. So what I would suggest is just to purchase our injection. It's different than micro injection. And that's the key. You can't even use our micro injection equipment for this stuff. You have to use a macro injection system. But like I said, it's cost effective. It's very fair. Yeah, so I would definitely use that.
Okay, thank you. In a newly detected area, what is the success in removing the tree to prevent spread?
I'm sorry. Can you say it one more time, Kendra?
Yeah, for sure. In a newly detected area, what is the success in removing the tree to prevent spread?
Um, well, it's interesting, and right now we don't know. However, there is some research. Once again, it's being done as we speak, and there's some trials that are being installed this year that are looking at in a forested ecosystem a combination of treating certain trees, removing certain trees, and seeing what effect that has on the overall population of beech. So right now, I have no answer for it. I don't know, but it's something we're thinking about and considering. I mean, if the tree's gone, you're not going to get it, I guess. But
Hopefully. Okay, and just last question here because we're running up on time. I joined a little late. Could you share information about the current geographical distribution of beech leaf disease as we know it?
Yeah, real quick, if you're north of Virginia and east of Michigan, I would be on the lookout for it.
Okay, thanks so much, Mark. And to those of you who we didn't have time to get to your questions, please feel free to email myself or Mark at this email address. And we're happy to carry that conversation with you. But for now, thank you so much for joining today. And thanks again, Mark. It was a great talk.
Yeah, thanks, everybody.
Have a good day. Bye.